Hello, my name is Inez Januszczak, and I'm the sampling coordinator for the Darwin Tree of Life project at the Natural History Museum. And I'll be talking about how we use DNA barcoding as an identity verification tool for invertebrate taxa. So an overview on the Darwin Tree of Life project. The project aims to sequence the whole genomes of 70,000 plus eukaryotic species in the UK. So this includes animals, plants, fungi, protists, and algae. And we work in collaboration with a huge number of partners listed in the infographic to the bottom right. So part of the project, although we are aiming for whole genomes, we are also generating DNA barcode sequences of identified species. So a DNA barcode is a short strand of DNA of a particular gene or genes, and is normally used to verify the IDs of a sequence specimen. The NHM alone, we generated almost 5,500 high-quality barcodes of mostly invertebrate specimens for the project so far. So DNA barcoding is key in verifying or not verifying IDs, but relevant taxonomic expertise is crucial in order to be able to do this as well. We also use DNA barcoding as a way of tracking the sample and quality checking the sample before it goes for whole genome sequencing. So how matches are made? For the Darwin Tree of Life project, we always identify the sample to a genus as a minimum. So from the sample, a leg or a relevant tissue part is extracted, and this PCR product is then sequenced. The reverse and forward strands undergo a QC filter, and they are then merged. So for the project, we only upload merged sequences, or we only used merged sequences for this analysis. CO116S are used, and these QC passed and merged sequences are then matched to their top 20 hits in online DNA barcode reference libraries such as BOLD, so the Barcode of Life database, and NCBI. So I'll be mainly referring to BOLD in this presentation, since that is the reference library that we work with the most at the Natural History Museum. And for us, an immediately verified sequence means that it has to match for 99% similarity and match for the majority. So we will only count a ID as being verified if it matches to this level on these online barcode reference libraries. So one issue is, of course, even if we are just talking about invertebrates, there is a huge range. So as you can see in this infographic, they will come in an enormous number of shapes and sizes, whether they have a hard outer shell, whether they have a mucus, their size, all of these have a huge impact in how successfully they are sequenced, and of course, how successfully we end up with a match. So how does the matching work? So here's an example of a very successful, straightforward match. So this is the rosy rustic. So as you can see with the rosy rustic here, it's 100% similarity across the board. 100% published sequences, so these are publicly accessible sequences. It's worth noting that although these online reference libraries are public and anyone can use them, some of the sequences are private. So we can see that across the top 20 matches, 100% similarity, 100% published sequences, we can verify this ID with total confidence. Here's another example of quite an iconic species, the Highland midge. You can see that it's more of a mix of published and private status sequences. However, still above 99% similarity. And because this is such an iconic species, we could be fairly confident with the ID despite this variation in data. So what does lead to a successful match? Well, like mentioned, if it's a well-studied species or it's common and widespread, you're more likely to have uploaded sequences, not just in the UK, but across the world. So this is a huge advantage for that species in itself. If it's distinctive and easy to ID, you're less likely to have misidentifications. You're more likely to have successful matches on these online reference libraries. If it sequences well, so some species just have better quality sequences, usually because they're more studied or better represented, such as within Coleoptera or Lepidoptera. So you're more likely to have matches within these groups compared to some others. Of course, we are detecting some interesting other results. For example, through this barcode matching work that we did at the Natural History Museum, we were able to verify a wasp new to Britain, so Plectiscus calidulus, which resulted from doing these matches and analysing the data and using worldwide references that have been available on these platforms. So we've had some really interesting results through comparing sequences from across the board. Now, when it comes to unsuccessful matches, Again, going back to well-represented species, so this is a hydroid and a telesecundaria. You can see here that the sequence is an absolute mess in that we have below 80% similarity across the board, as well as a huge range in species and families, despite the fact that these are mostly published sequences. So there's a huge number of reasons for this. Of course, a hydroid is a colonial animal, so there could be a case of multiple organisms in one place, which would be harder to pick out using DNA barcoding. Misidentifications, poor quality sequences. Reasons like this mean it's even more important we have a taxonomist on board who is able to verify the ID, not relying just on DNA barcoding data. Also, it's very common that we have examples such as this. So this is a nudibranch where it was originally ID'd as Polycella quadrilineata, but as you can see, that only appears once in our top 20 hits. There is a huge range in species matches. Now, the sequence in itself, we can assume to be of fairly good quality since it's above the 99% match. However, we will still need to look at the source of the sequence and the source of the identifier, since clearly there has been cases of misidentification here in that it is matching so highly to 2 or 3 species within that family. With unsuccessful matches, the case is usually that the species is rare, the species is hard to ID. If it's cryptic, even if it's common, that means it could be easily misidentified as well. A huge lack of expertise in some orders is also a big problem, less people to verify these sequences to start with. If it sequences badly due to the condition of the specimen, due to the fact that there's mucus, it's been stored incorrectly, then this also decreases our number of matches. Misidentifications just generally are a problem across the board, as you saw from the last example. And finally, if it's new, if it doesn't exist in the reference library in itself, then that will be problematic and that you will get very poor matches regardless, because it's the first time that this barcode has been generated for that particular species. So just as an overview from the Natural History Museum, our last 10 barcoding plates, we had a 77.7% complete match, 14.5% fail, and 7.77 pending future identification. So fail basically means that it did not sequence at all. So this is a decent set. However, obviously, ideally, we would like to improve the number of matches that we get and decrease the number of failed sequences from the start. So moving forward, from our perspective, we think it's crucial to only upload verified high-quality sequences to add to these libraries, or you are simply just adding to the number of sequences that are not high quality or misidentified, which then just perpetuates the problem. It's crucial that we share taxonomic expertise, encourage taxonomic expertise to have these sequences verified by the relevant experts. And it's incredibly important to use a combination of the sequence data as well as morphological data to ID these specimens. Hence, the danger in relying solely on DNA barcoding for this kind of work means that you could be populating these libraries with misinformed or misidentified sequences. Thank you very much. I would also like to thank Dr. Gavin Broad, who's the co-principal investigator of the Darwin Tree of Life project at the Natural History Museum.